All right, we are back, everybody. Welcome. Welcome back to our next video, our next research topic here. So today we are going to be talking about three-dimensional organoids. What is an organoid? How do we use it? And in particular, this study we did with pancreatic organoids. So can we use these pancreatic organoids to predict response of a tumor to radiation therapy before it has radiation therapy? So that's what we're going to talk about today. Sounds like good stuff. So as always, my name is mediphysman, and these are my medical physics lectures. So today's topic: can 3D organoids predict the response of pancreatic cancer to radiation therapy? Can they? I don't know. Maybe. Let's find out. So, let's start with our the legal stuff. Disclosures, disclaimers, conflicts of interest. Really? I don't really have any. Nobody's paying me to do this. Nobody's advertising on these videos. Just me talking about research. We did receive a small seed grant from the Department of Radiation Oncology at the University of Maryland School of Medicine to study this idea. So thank you to them. That was great. Let's talk about our collaborators. Tiana Dukic, Binny Bandari, Sanjeet Roy. They're the ones that do the real work. They're the ones that actually do the work in the lab. Let's be honest here. I'm going to put that out there to begin with. Tiana is our research associate. Binny is the lab manager and our senior researcher. And Sanjeet is our postdoc. I also work with Dr. Him Shukla. He's the organoid expert. He knows how to make organoids. So he and I work together making organoids, then treating them, seeing how they respond, having lots of fun. Naru Lamanchain, he's a good friend of mine. No matter where you are, he's always the coolest guy in the room. He keeps us on track, keeps us focused, keeps this whole project moving forward. And then there's me, of course, mediphysman. I'm the crazy idea guy. You know, I'm full of crazy ideas. I tell a good joke. I think for that reason they keep me around. That's really the main reason. And as always, let's talk about this. Please, if you like this video, please give us a like. Please subscribe to the channel. The more likes, the more subscribers we get, the better conversation we can have about science. We can put out more videos about more interesting research. We can discuss it openly together. We can learn from each other. We can have fun. I mean, that's the whole point of what we're trying to do here. Let's come together. Let's talk science. So, for this study, what is an organoid? We're going to talk about that. We're going to discuss organoids. We're going to discuss the experiments we did with organoids. Then talk about the data. What do they tell us? Can we predict treatment response? Does this data tell us that? And then talk about what we learned. Okay, what did we learn? Where can we go from next? What can we do with this stuff? So let's start with our background. What is an organoid? I don't know. Let's find out. So what we do with an organoid is we first take cells, we biopsy them from a tumor, from a mouse tumor, from a human pancreatic tumor. So we get a biopsy from the cancer here in the pancreas. We take and we digest that tissue sample down into individual cells. So we digest the cells apart. You get cancer cells, you get fibroblasts, you get stellates, you get all kinds of cells that are there within the environment of the cancer, all within your dish. We then culture those, co-culture all those cells back together within some matrigel within a dish, and lo and behold, those cells will start to self-organize and grow together into something that resembles a pancreas and resembles the pancreatic tumor. The shape, the morphology, the heterogeneity of the cells, how they all interact with each other, begins to mimic what we would see in an in vivo tumor. And people have done lots of studies with this. Lots of organoids different types of organs, from the brain to the kidney to the heart to the lung, you name it. They take and they grow these organoids, and we've developed a large number of different types of what are called microfluidic chips. So we can grow what we call a tumor on a chip. Others call it an organ on a chip. And they have these channels where you can seed and grow the organoid. You can then flow nutrients and blood and different treatment agents through there to see how the organoid responds. And they've done some amazing work with this. This is a picture of a beating heart. It's an organoid of a heart that's begun to self-organize and start to beat like a heart does. That's just amazing to me. So within these microfluidic chips, within these organoids, what we can grow is something that reproduces what you might see in a pancreatic tumor. So you have this center necrotic region, you have a oxygen starved region, not quite necrotic, and then this normal oxygenation region outside of it, and all that's grown in the center channel, and you flow nutrients and different culture throughout it through this channel. And what we get then is something that starts to organize, difficult, it's hard to say. Anyway, organize itself into a pancreatic tumor. Yeah. So with this, we can then study how it grows, how the cells organize themselves, what the microenvironment looks like, and how all of these things change in response to treatment. So organoids are something, like I said, and cancer organoids, tumor organoids, have been studied widely for the development of new chemotherapy drugs. This is something that's done extensively. And so if you do a search online for papers published with the keywords chemotherapy, tumor organoid, or patient-derived tumor organoid, you get something like 12,000 or 1,200 papers, well over 1,000 papers published on the use of organoids to study chemotherapy. But if you look in radiotherapy, on the other hand, yeah, what do you think? More? Less? More? Less? Well, radiotherapy, not so much. 52 papers, that's right, 52 in the all-time history of organoids. 52 papers. So, not studied, they're not used a lot for radiotherapy. And we were just wondering, why not? Why can't we use these wonderful things to study response to radiation, right? And so that's the whole genesis of the idea we wanted to study here. Can 3D organoids predict tumor response to radiotherapy? So, our experiments. What did we do? Okay, so we took tumor tissues from a pancreatic tumor that was grown in a mouse. We took those tissues, we digested them out, we co-cultured them together, and grew a large number of organoids. So from a single tumor, from the tissue harvested from a single tumor, we could grow an extremely large number of organoids. So much that we could divide them into 4 separate treatment groups. So we studied no treatment, that's our control. We studied X-ray radiation, so tumors, mouse tumors, and the organoids that were derived from those tumors. We studied those for X-ray irradiation. For chemotherapy with 3-bromopyruvate chemotherapy, and then with X-ray plus chemotherapy radiation. And so from that, we then studied how the tumors grew after treatment. So we studied how the tumor in the mouse grew and how the organoids grew over a course of 10 days. So if you want to know how did we grow the tumors in the mice, here's a breakdown of how that was done, and then how the tissues were harvested from the mouse. And then how do we remove them, how do we digest them, and how do we make organoids out of them. It's all right here. If you want to read this, feel free, pause the video, get a good sense of what all's going on here. You get the full recipe. After we did all that, how did we study these organoids and these tumors? So we did H&E staining and imaging of the in vivo tumor tissues as well as the organoid tissues, as you see here. We also did immunofluorescence staining and imaging of the organoids and the in vivo tumor tissues, and you can see how that was done here. Next, how did we do irradiation and treatment? Well, here's how we did. Here's the irradiators we used, and our dosimetry was all done according to the double APM task group calibration protocols. This is what we did for the mouse tumors. This is what we did for the organoids. Tried to irradiate them in the same manners. And then how we treated them with the 3BP chemotherapy agent. And then how did we evaluate them afterwards? So we took the evaluation of our mice, of our in vivo tumors, and our organoids. As you can see, for the organoids, what we did is we put a square around them in the image, and we took the area of that square, and we used that as to mimic or to study how they were growing after treatment. So, as you can see, here's our organoids growing in a dish. Day 0 when we start. Day 6, you can see they've grown much larger. They're starting to grow together into one large blob. They're sending out these fingers and tentacles throughout the dish. And then by day 10, it's grown enormously. It is this big blob that's got these tentacles going out. They're connecting with other organoids in the dish and starting to all grow together into one large organoid. Same with the H&E staining. You can see just a blob here. Even on day 0, you can see it's already started to send out these tentacles. You can see how they grow outward extensively and how the cells start migrating around off of those tentacles, and then how it all grows together into this one large, big tumor organoid. As far as the microenvironment goes, what we have here in A and B, we have the in vivo tumor tissue from the mouse, tumor from the mouse. We also have an H&E image of the organoid tissue, and you can see how they're both starting to grow and develop this fibrotic nature of how the cells are organized. Again, very hard to say. And then we do the fluorescence imaging. You can see we stained for fibroblasts. So the fibroblasts will show up with the immunofluorescence when we do the imaging. You can see a large number of fibroblasts within the in vivo tumor environment. And then the same in the pancreatic organoid. You can see a lot of fibroblasts and a lot of green fluorescence from the fibroblasts within those tumor organoids. So they both seem to have the same sort of fibrotic environment and the same types of cells contained within both. Okay, so how did our organoid respond to treatment, and how did that compare to the in vivo tumors? So here you can see our in vivo tumor in the mouse. So that tumor was treated. So we have the control, the untreated one. You can see it kind of grows exponentially over 10 days. And the same with the in vivo, or with the organoid. The control untreated organoid also shows the same exponential growth over 10 days. However, when we treat them with 8 gray of radiation, and we look at this growth factor, right? So our growth factor is the size at day 0 divided by, or the size on a given day divided by the size on day 0. So day 0 growth factor of 1. If it's growing bigger, the growth factor is bigger than 1, right? If it's shrinking, the growth factor is smaller than 1. So control, they both had exponential growth. 8 gray of radiation, so we treated it, gave it 2 gray once a day for 4 days, total of 8 gray. And you can see we've almost completely stopped growth. Growth factor is right around 1 for both the in vivo mouse tumor and the organoid that was grown from that tissue. So if we look at the cellular response, how did they respond to irradiation? Here we have the in vivo tumor before and the in vivo tumor after, right below that, are on the left. You can see the H&E image. And if we stain for cleaved caspase 3 biomarker, so this is a biomarker that's expressed when the cells enter the apoptosis cellular death pathway. And so before treatment, we don't see any signal for cleaved caspase 3. So really no apoptosis going on in that tumor. However, the tumor we looked at after treatment, you see the orange staining show up. There's a large number of cells that are undergoing apoptosis after treatment. And we see the same trend in the organoids after treatment. So before, no cleaved caspase showing up. However, afterwards, we see a large amount of fluorescence indicating the presence and of cleaved caspase 3 meaning a lot of apoptosis going on in the cells after treatment in the organoids. So we see that our cells, our tumors sort of grow and have the same microenvironment, and they seem to be responding to radiation in similar manners. And so if we look here, we know that it seems like our pancreatic organoids give us a good model. They're a good model for what's going on in the tumor, at least for irradiation as we've seen here. So if we look at how it responds to different doses of radiation, so we see here day 0, day 6, day 10, our squares around it are getting bigger, right? And then we look at 4 gray, 6 gray, 8 gray. We can see, and here we have 5 different organoids that were followed over 10 days, and you can see their growth factor. And the black line is the average of all 5 of those. So when I show you numbers, that number is the average number, right? So you can see an exponential growth for control. It flattens out for 4, flattens out more for 6, and then for 8, pretty much completely flat. So we think we've done a good job of stopping growth with 8 gray of radiation. If you look at this in terms of the growth factor values, you can see untreated, we have a growth factor of 10, 10.6, 4 gray, 3.6, 6 gray, it's gone down to about 1.5, and 8 gray, it's right at 1. We have a growth factor of 1, right? Again, getting bigger, but not so much for the 8 gray. If we look at this with the bar graph, I don't know, I like bar graphs, lots of pretty colors, I think that's why. But you can see that exponential growth, and our error bars here on the bar graph represent the standard deviation of all 5 of those organoids that we followed. So what's the growth factor for all 5? This is the standard deviation. Again, the bar value is the average of those 5. So now, what if we treated our organoids with chemotherapy? So here's the 3-bromopyruvate targeted molecular treatment drug. So we gave our organoids 100 micromolar concentration of 3BP. We also did a 4 gray irradiation, and we also did 3 BP plus 4 gray irradiation, right? And so here's our 5 organoids we followed for each one. Again, we see that exponential growth. It flattens out at 4. It's pretty much flat for 3 BP, and then for the 3 BP plus 4 gray, an ever so slight dip. It's actually a small decrease of that line. And again, here's the bar chart, exponential growth, and it flattens out and gets, a tiny bit smaller for the 3 BP plus 4 gray. You can see that here with the growth factor. For the 3 BP plus 4 gray, we had a growth factor of less than 1. These things were shrinking ever so slightly, starting to dissolve away. So we've really done a good job. The combination of the two is actually completely taking care of the tumor cells, and the organoid itself is not able to even sustain its own shape and size. Okay, so this is fun. So this is a movie. We were able, with one of our microscopes, to get an actual movie of the organoid growing. So here's our untreated control organoid growing over 10 days. So it doesn't start at 1, unfortunately. I don't know why. Okay, here we go. Day 1, it's getting bigger, bigger. It's other things growing that kind of absorb each other into one bigger blob, and the morphology's growing. It's drifting around. It's pretty cool to watch. I kind of like it because you can really get a sense of what's going on with these things. So what about the 3BP plus 4 gray? Well, here's a movie of that one. Again, it doesn't start on day 0. Let's let it cycle through and we'll see it at 0. Okay, here, day 0, it's growing, or it's there. It's not really growing. It's not changing. The morphology is the same. It is drifting across the plate. It's interesting how they're both kind of drifting across our petri dish through the matrigel as they grow. I don't know if that's a factor of the microscope or what, but it's interesting that they're moving. Everything seems to be moving. But anyway, I think our untreated control kind of reminds me of the blob. It eats everything. Blah. Did you ever see that? It's that 1950s sci-fi movie. It's pretty campy. It's fun. But then the other one kind of, to me, reminded me of like a planet transiting the sun. You know, it doesn't change or grow or anything. It just kind of moves across and you see it there. It's not much going on, but I mean, it still looks really cool. I like watching it. So anyway, this is from Wikipedia. the video from NASA. That's Mercury transiting the sun there. And the NASA video is pretty cool. All right, so what do we learn from all this? Well, we've developed this pancreatic ductal adenocarcinoma, PDAC, 3D organoid model. We're still developing it. We're still perfecting it. So we can reproduce the cellular microenvironment and the heterogeneity found within the tumor that we see in vivo tumor. We can reproduce that in our organoid. Again, we're still engineering that and trying to perfect it to make it reproducible, make it stable, make it the best we can. It's interesting, we can make organoids from normal pancreas tissue. We can also make it from pancreatic tumor tissue. And you can see the difference. The normal tissue makes these nice, uniform, spherical organoids, right? Where the pancreatic tissue is just this massive blob that's just expanding and reaching out everywhere. We studied our pancreatic organoids. They showed the similar growth rate and response to radiation as the in vivo tumor did. So that's good. We think that it turned out to be a pretty good model to predict response. And we showed that you could study this organoid and its response to other treatment types, increasing the dose, using chemo or chemo plus radiation, and see which one did the organoid respond best to. So there's a lot we've learned. Of course, there's a lot of work that needs to be done here. We're just getting started, just scratching the surface, right? So what's next for us then? Everything I showed you today here, these have all been 2D images using our bright field microscopes, but really these are three-dimensional objects. So we want to study them in 3 dimensions. So we've started studying and looking at 3-dimensional, like confocal microscopy, other types of 3-dimensional imaging, to look at the shape and morphology of the organoid in 3 dimensions. As you can see here, this organoid, as you go through, the morphology changes a bit, quite a bit, changing in size and shape. And when we stain it here, these are for live cells, different types of cells. You can see how the number and how they spread out, the location of these different types of cells changes as you go through the organoid. So we want to look at this 3D imaging to study volumetric growth, how the microenvironment evolves and changes, and how cells migrate throughout them, and study things we can learn in 3 dimensions and even 4 dimensions of these organoids. What's the best way to assess how they're responding to treatment? Can we use this sort of stuff? So that's what we're looking at right now and doing more studies on. The long-term goal overall, what we would love to see with this, what if you could take a biopsy of pancreatic tumor biopsy from a patient, you digest it, produce a whole series of organoids, and you expose those organoids to a range of different treatment types, chemo, radiation, combined, different regimens of whatever. We grow and we assess how those organoids respond. How do they change? How do they grow? How does the microenvironment evolve? And from that, we're able to predict, oh, look, your tumor responded best to this chemo-radiation combination. You had the lowest viability and best cell killing within your tumor. So this was what we would treat you with. If somebody else had a different tumor and it responded differently, and say it responded best to standard radiation therapy, then that's what they would get. So you could use this as a tool to try to personalize how they are treated for their pancreatic cancer. So, all right, I'll stop talking. I'm going to stop talking here. That was good. So if there's any questions, please leave those down below, and we can start a conversation here, right? Let's talk about this. Please tell me anything I missed in my data. Did I misinterpret something? Is there something more there? I want to learn. I love to learn. I want to learn more from you guys. Let's see what we can come up with together. Let's have a good conversation. That's what it's all about. Let's talk science, And as always, please like and subscribe. The more you do that, the more videos I can make, the greater, the more fun we can have talking science together. All right? Thank you very much, and I will see you on our next video. Okay, I'm back, guys. Just a quick disclaimer on this video. We talked about studies using mice in the research. Personally, I'm not a big fan of that. I don't like animal testing. The idea of using animals to test stuff. To me, I have an ethical problem with that. I don't like it at all. The data I showed you involving mice and the pancreatic tumor tissues we got from mice, this was a study that was ongoing in another lab. Another set of researchers, they were doing this. It's funded by people and they do their own research, and that's their thing. Everything they do is under strict approval by the university for usage and handling of the animals. Everything is done according to the book, and they're really good about it. Personally, I don't ever want to be the person involved or in charge of an animal study, because I don't want to have that on my conscience. It's not me. I could see how the work is necessary. I don't know. It's I've seen good results that have gone on to help people come out of these animal studies. I mean, someone does an animal study, it results in a breakthrough drug. That drug can be given to kids to cure their cancer, and so now all these kids get cured from their cancer. Is that a bad thing? That's a good thing, and I wish the animals didn't have to be involved, but thank them so much that they helped us. Anyway, animal studies, not for me. We got our data from another study that was ongoing, and we were able to use that on the organoid side. So really the only thing I had a hand in were the organoids themselves. I didn't want to be involved in the animal stuff. That's what made this possible. Maybe an ethically gray area. I used tainted data from animals over there, but at least I wasn't the one responsible for them, and so it makes me feel a little better. I don't know. I'm not condemning anyone who does animal studies. Like I said, I can see where they're necessary and where they've done good things. Just for me personally, my own personal opinion, my own personal feelings, I don't want to be the one that does them. So anyway, I just thought I needed to put that at the end of the video to help me, at least help me feel better. Again, something we can discuss, in a nice, calm, appropriate, professional manner in the comments if you'd like. But either way, that's my thoughts. Thank you.